Good morning, everyone from Providence, Rhode Island. It's my honor and pleasure to start the Israel, Rhode Island, United States Blue Revolution and Sustainable Aquaculture Conference. My name is Avi Nevel. I'm the founder and the CEO of the Rhode Island Israel Collaborative. We are a non-for-profit chamber of commerce that connects academia, business, and research. Before I go any further, I would like to thank our co-host of this program. First to Dr. Galit Sharon. She's Associate Professor of Biology at Roger Williams University. Galit and I met more than a year ago, and we started to dream this conference. And I have to say, finally, we got here. Galit, thank you so much for all you do for this project. I want to thank also Karen Almozlino from Aquatech, the community and manager of Israel Innovation, and Ronnie Sussman, the CEO of Aquatech, the Israel Biotechnology and Aquaculture Community. Both are from Eilat, Israel. Many thanks also to Roger Williams University, who supports this program. I have to say that this is a great example of collaboration and continued collaboration between the Israeli ecosystem here and in Rhode Island. Last month, we had an event in the subject of AI and art with RISD University here and people from Israel. And what we feel and like that there is ongoing collaboration. And today we are working on the subject of Blue Tech and Aquatech. But before that, I would like to go and thank all the supporting organization here in Rhode Island and in Israel. And by reading them, you will see the depths of the collaboration and the support. So as I mentioned first, Roger Williams University, Commerce RI, the Hispanic Chamber of Commerce of Rhode Island, Innovation Newport, Rhode Island, Newport Chamber of Commerce, Greater Providence Chamber of Commerce, the RI Hub, and RI Bio. And thank you for the Israeli organization, Israel Consulate General in Boston, Israel Economic Mission to North America, Israel Innovation Institute, the Innovation Authority, Ministry of Economic and Industry in Israel, and the Ministry of Agriculture. And more importantly, thank you all who participate and to the speakers in this event, because you are the ones that will give it a content and importance of the collaboration. So as I said, there's no doubt that the collaboration is strong, and I'm sure it will be stronger. Please remember that this is the prelude to our June 3rd and 4th, 2024 in-person conference on the subject that will be at Roger Williams University here in Rhode Island. A couple of announcements and requests. One, please mute your microphones. Two, if you have any question you want to raise with the speaker, please type in the chat box in Hebrew and English, and the moderator of the session will choose one question. We will be more than happy to connect you in this conference with any specific speaker or any specific participant. Fourth, to the speakers, please try to adhere to the time, as there are many speakers. Presentation would love to give everyone the time that's allocated to them. We will record this program and we'll share it with everyone who will be interested. Last, before I hand the microphone, I would like to thank to our dignitaries who sent recordings. Liz Tenner, she's the Rhode Island Secretary of Commerce, Rhode Island Lieutenant Governor Sabina Matos, Anat Katz, Israel Economic Minister to North America, and in person, Brian Williams, the Chief of Staff of Roger Williams University. With that said, I would like to invite my colleague to take over and continue with the program.
Thank you, Avi. Hello, everyone. I'm Karen, Community Manager at Aquacaltech, and we are one of 7 communities at the Israeli Innovation Institute. And I'm going to share our story with you in a short video.
We face so many global challenges: urban mobility, food crisis, better and universal healthcare, desertification, climate change, and ocean health. We need to develop new technologies and find better solutions to make this world more sustainable and increase human welfare. The Israel Innovation Institute works to harness the innovative activity in Israel as the startup nation to lead the world in addressing these global challenges. Our targeted innovation communities, EcoMotion, Health IL, Growing IL, Catalyst IL, Desert Tech, Planet Tech, and Aquacultech are actively involved in shaping their sectors through knowledge sharing, best practices, funding, and creating effective collaborations, promoting open innovation, and implementing innovative solutions. Israel Innovation Institute channels the Israeli ecosystem to global challenges and helps it to act better and faster, transforming Israel from a startup nation to an impact nation. Israel Innovation Institute, where global challenges become innovative solutions.
Please, Ronnie Zussman, CEO of Aquacultech.
Aquacultech is one of the 6 communities of the Israel Innovation Institute, and we are developing the ecosystem of biotechnology and aquaculture in Israel. Our first mission, when we just started on February 22nd, was to map those that are active in aquaculture in Israel. And from there on, we are active in creating networks between people, between all stakeholders in this sector. And we are supporting all startups that are active in this area of aquaculture. Aquacultech is now with 1,300 members. Most of them are from Israel, but we do have partners abroad. And we are here in this event, like any other event that we are creating from the community, according to the request from the members of this community. We had industry academy events, we had investors events, and we had locals events for development of the Eilat area that we are located in. And we are supporting the governmental program that invests 170 million shekels to develop the Eilat area to be a knowledge hub for this sector in Israel. Any connection with any company from Israel, for any connection, you are welcome to connect with us, and we can create the meetings or just answer any question. And that's it. Enjoy this day. Please keep your computer muted, and we promise to attend any question.
Good morning, everyone. My name is Liz Tanner, Secretary of Commerce for the state of Rhode Island. It's a pleasure to join you all for today's Blue Revolution Sustainable Aquaculture Conference. Today's conversation is an important one because Rhode Island is home to a wide range of maritime industries, such as boat building and servicing, the largest independent steamship agency in North America, submarine building, the development of advanced materials like processing components in boat and ship systems, preservation of coastal communities and environments, and a coveted tech hub designation from the Biden administration, catalyzing local innovation in ocean sciences and engineering. Rhode Island is ready to build on our highly integrated maritime economy that encompasses manufacturing, research, and reinforces the competitive advantages of the ocean state. Here in Rhode Island, aquaculture plays a big role. Rhode Island has 84 individual aquacultural sites, representing a combined 370 plus acres and more than 240 aquaculture farm workers. The combined value of aquaculture products for consumption and seed sales in 2022 was over $8 million. We sold over 10 million oysters for consumption alone last year. That's a lot of oysters. Over the past 10 years, the total number of aquaculture products sold, the number of farms and acreage under lease, and the number of workers employed in the industry have all steadily increased. That tells us there's growing demand for this kind of work. We are also committed to ensuring that Rhode Island's diverse marine ecosystem continues to thrive for generations to come. We are working with the USDA Natural Resource Conservation Service to purchase and deploy over 1 million oysters to restoration sites throughout coastal ponds and the Narragansett Bay. Now is the time. If you are a business looking to capitalize on Rhode Island's competitive advantages and our existing marine infrastructure and new ocean tech hub, we want you to consider growing your business in the Ocean State. I want to thank Avi Neville for his leadership on this important issue, and I want to congratulate the Rhode Island-Israel Collaborative for winning an award at last week's Go Global Awards, hosted by the International Trade Council and Rhode Island Commerce. I look forward to seeing all that comes of this important work started here today and what lies ahead for the rest of the conference. Have a great rest of your day.
Good morning. This is Lieutenant Governor Sabina Matos. I'm so excited about this conference. The exchange of technology and experience between Rhode Island, Israel, and Greece makes all of us much stronger. Now more than ever, we need the cultural connection between our nations. Hosting economic and scientific forums like this reinforce our steadfast support for Israel. By sharing ideas that are important to us and our goals for the future, we strengthen the bonds between our nations. It is my hope that we can continue to grow and make our state a global hub for researchers and scientists. Rhode Island was the birthplace of the American Industrial Revolution. We want to lead the way once again. Our waterways fueled that Industrial Revolution by powering our mills. Now, they are ready to fuel a new blue revolution. Our strong partnership with the global aquaculture community is just the beginning of that work. We also want you to know that Rhode Island is open for business. If you have an idea, we want you to execute it right here. Please reach out to my office. We would love to help you set up shop in Rhode Island in any way we can. Let's work towards a future where sustainable aquaculture can support all of our nations. Thank you to Rhode Island Israeli Collaborative and Roger Williams University for hosting this informative conference. I wish you a fruitful day of learning and sharing. Thank you.
Hi, everyone. My name is Anat Katz, and I'm the Israel Economic Minister to North America. The Government of Israel Economic Mission is physically located in New York and basically caters for the entire eastern region of the United States. I want to open by acknowledging the fact that these are very challenging times for all of us, clearly for everybody in Israel and for many of us here, either Israelis in their homes away from home here in America, and other members of the community. I want to thank everybody for joining us today, not just because this conference showcases Israel, and not just because of the fact that I know that for some of you, the actual participation in this is a show of support, of which we're greatly thankful for, but actually because of the topic of this conference. The topic of this conference basically gives us a platform of reiterating who we are and what we stand for. We stand for innovation. We stand for sustainability. We stand for life. Our economic mission works to connect Israeli entrepreneurs with the local ecosystem in multiple sectors. So it's really everything from food consumer products to healthcare, cyber, fintech, food tech, and everything in between. So basically really hummus to healthcare, everything. We collaborate with all the organizations that basically see eye to eye with us in this mission. And it is for that reason that this invitation from the Rhode Island Israel Collaborative was extremely right down our alley. And I was happy to accept, and I'm thankful for Avi for inviting me and for including me in this important session. The aquaculture area and sustainability are actually part of a national program that we have for economic development. This is a program by our Minister of Economy and Industry, Nir Barkat. Basically, at the heart of this program is 6 industry clusters. One of them has to do with aquaculture and sustainability, and will be basically located and based in the southern part of Israel, especially in the city of Eilat, for those of you who know it. Part of the program is funding for joint research and development initiatives, as well as for academic collaborations. So I invite everybody to join us in this national program and to participate and foster and work actively in collaboration in these sectors between Israel and the United States, and specifically Rhode Island. I want to take this opportunity and thank the Secretary of Commerce, Tanner, as well as Governor McKee's administration for supporting the bilateral initiatives. I want to extend a specific thank you to Lieutenant Governor Mados for joining us today. And last but not least, I want to extend a huge thank you to Avi Neville, both for setting up this amazing event, and really for everything that he's doing for our communities, both here and in Israel. By bringing together the brilliant visionaries from both sides, I think that we are definitely actively promoting the realization of the future of innovation, of sustainability, of growth, and of peace. I want to thank you all for joining today, and I'm wishing you a very, very successful conference. Thank you very much.
And now we'll have Brian Williams. He's a chief of staff of Roger Williams University. And as I say, they're our key partners, and we really appreciate to that opportunity to collaborate. Brian?
Great, thank you so much. On behalf of our president of the university, President Yanis Mialis, who's from Greece, want to welcome to this virtual session as a preface and a taste of what we'll be able to do together this summer. You've heard a little bit, Rhode Island is really a hub for the blue economy and the blue revolution. And our nickname in the US is the Ocean State. So we really try to be a coastal and ocean university committed to that space as the state moves. Our hometown in Bristol is really a nexus for so much of the blue economy. We are located right on an amazing estuary. Our aquaculture faculty are key to the industries in and around us since the 1990s, when they were founded over 25 years ago. And we are a hub of sailing boat manufacturing. Rhode Island is one of the first areas of offshore wind development in the US. And there's a large push towards blue tech innovation, underwater autonomous vehicles and robotics that's really pushing in the state and in the city right around us. So those opportunities are strong. We are really passionate about this intersection of academic research and industry partnership, that our research that's done here moves industries forward. And you see that in our Center for Economic and Environmental Development. This conference really excites us for the opportunities that it has. And beyond that, across this university, you have a School of Law that's passionate about marine affairs and maritime law. You have policy folks that focus on coastal development in our School of Architecture and School of Engineering, and think about building resilient communities. So we're really excited that we think we're a great backdrop for this June conference. I can't wait to meet all of you in person in June and to hear your ideas and learn from you all together. Bringing together and being a convener is something we're passionate about. I'm excited to learn from you today and excited to do that in person in June. So thanks for being with us, and look forward to the other speakers' remarks through the rest of the day today. Thanks.
Thank you again, Chief of Staff Brian Williams. And now we're going to ask Dr. Galit Sharon to speak and take over the program. Again, in June, we'll have the conference in person. We'll announce it with the details and registration. And we hope to have also other countries as we want to open it as wide as possible. So with that said, Dr. Sharon, please, it's yours.
Hello and welcome, everyone. My name is Dr. Galit Sharon, as you heard. I'm the Associate Professor of Biology and the Director of the Aquatic Diagnostic Lab, which we call the ADL, which is a part of the SEED. So I mentioned that a little while ago. So the Center of Economics and Environmental Development here at Roger Williams. The ADL is the Aquatic Diagnostic Lab, where we engage in extension work and with aquaculturists and farmers throughout the East Coast. And we do different diagnostic methods to detect diseases. But we also do research, and our research projects are in fields of development of new methods of diagnostics, studying pathogenesis of different animal emergent diseases, different aquatic animals emergent diseases, and development of innovation of treatments and management methods. So that's a little bit about where I come from. And I'm excited to have you all here. I'll take just one more moment of your time, and I'll invite you all to the Blue Revolution Sustainable Aquaculture in June in the Bristol campus in Roger Williams University. During this event, we will hear more researchers and businesses from different countries that will actually talk about their work. And then the second half of the second day, we want to do a roundtable discussion, meeting and interactions between participants in order for different collaborations to come alive. So we will have, like Avi said, more information, and we'll share the registration and submission of abstracts with you. So with no further ado, I want to introduce the first speaker, Professor Andrew Ryan. Professor Ryan is a biology professor and a researcher, and one of the first and main faculty in SEED, the Center of Economics and Environmental Development at Roger Williams University. Professor Ryan will talk about bringing academia and industry, aquaculture innovations at SEED. Professor Ryan, you can share your screen. Please don't forget to de-share the screen after, so the next participant can talk. Thank you.
Thank you, Galit. Thank you for having me. It's nice to see everyone. Some of the work that we do, specifically, and how that ties in with aquaculture and innovations in our work here. And so I'm going to give you a quick overview of a little bit about SEED, and then I'm going to talk so that I am deeply involved in, just to give you kind of an idea of where we're at. But I was brought here, and I started in 2008 through a joint partnership between New England Aquarium and Roger Williams University. And our group has always been really innovative and creative. And Tim Scott, our longtime director of SEED, created this collaboration with the New England Aquarium, which brought me up from Florida to create a joint program in aquaculture and aquarium science through a National Public Aquarium and a 4-year undergraduate institution. So it's a unique program in the country. And so when I look at aquaculture, there are many sectors of aquaculture, whether it's biotechnology or food production, restoration aquaculture, or my area of focus, which is aquarium aquaculture. But they really have very similar links to them. When you're raising animals, they require more or less the same technology, no matter what your end use is. You might have different goals, and you might have to have different conditions because of those goals. But the technology we develop in my area is easily transferable to restoration, or to food aquaculture, or to biotechnology or nutraceutical development, or other types of development. So there's a lot of cross-disciplinary work that can occur when you're working in one of these segments, as long as you're connected to the others. And so SEED was established in 1997. It's a very unique organization inside of at the time, a small liberal arts university here in the United States. So we only had undergraduates. We still only have undergraduates in our division, and it really was developed and fostered to drive collaborative marine research, to support industry and marine education, and then to engage in public outreach engagement and extension. And we have done a tremendous amount of that through the years. Primarily, the work had been on fish and shellfish aquaculture when Seed started. And Tim built this operation up from a very small, a large amount of funding to build a new hatchery for the shellfish hatchery. And so we still work heavily in shellfish, but fish, but we've also added other aspects of things that we're working on that really support the blue economy, and also we're really focused on sustainable aquaculture. And we at the time we housed the only shellfish hatchery in Rhode Island. This is the Luther H. Blount Shellfish Hatchery, and we have a very capable hatchery. We have a new faculty hire in that hatchery that is focused on shellfish larval work. We have a experimental farm that we've hired another faculty member, Hisham, whose job is to work in the field and extension work, and we have a whole team of folks that work in the farm and work with Rhode Island aquaculture producers of oysters. And on it to us, and again, this is the Aquatic Diagnostics Lab is an outgrowth of Tim's leadership and ability to grow our program and think about what was needed in the area. And a huge need for aquaculture is diagnostic services. You're producing seed or animals and you want to move them across state boundary. Rhode Island, very, very small. And it's really, I joke, and it's actually true, that I drive to Massachusetts to get ice cream with my kids. But if you were to take shellfish across the state lines, you actually need a permit, and they need to be certified to be able to do that, even though literally I can look out the door and I can see Massachusetts across the water. So this was a really big add-on to our program. And we brought Dr. Roxanne Smallwicks into this, and she kind of built the ADL, and then she recently retired, and we hired Galit, and we're really excited to bring Galit's capabilities and interest into our program and continue to grow this area. We also have a coral microbiologist in our group, Dr. Cody Sharp, and she works with a local coral. It's actually the state coral now of Rhode Island. It's a temperate coral, looking at all kinds of issues with coral husbandry and microbiology and biotechnology development. But for my part, I'm really focused on aquarium science and aquaculture. We have about 25 to 50 undergraduate students in the lab every year. And we have a lab that's very capable for developing new aquaculture methods for species that are popular in the aquarium industry. Part of that industry is the aquarium hobby industry. And I know from my decades of work with trade data and looking at imports in the United States, Israel actually has a very vibrant production of aquarium fish. I've known about several of the companies in Israel for many years because they export a lot of really high quality fish to Europe and to the United States. So we work in the aquarium industry that you would know as people's fish tanks, private homes, but we also partnered up with the New England Aquarium. We have our own exhibit at the New England Aquarium that displays fish that we raise here at Roger Williams University that are then distributed across aquariums across the country. And that has become a program that's not just Roger Williams and New England Aquarium, but has become a national program across AZA. And so we've started a cooperative program with aquariums, and we've really trained a lot of aquariums across the country how to raise animals and raise fish for their own exhibits. And we've started a cooperative, so institutions are raising animals and then sharing them with others. And that has become an official program within the Association of Zoos and Aquariums. And part of that program is we've been working to help us identify all these eggs of fish that are spawning on exhibits. So this is what a public aquarium will look like often, these beautiful large tanks of multi-species exhibits. This is at the Steinhardt Aquarium at the California Academy of Sciences in San Francisco. And fish are well fed, they're well taken care of, and so there's a lot of reproduction that happens in these tanks. And you collect the eggs out of these exhibits, and you really get a smorgasbord. You have no idea what you've got. Right. And so when you're trying to raise something, rearing fish with an assembly of eggs can be really challenging. And so we have been working on techniques to barcode these, and we use egg morphometrics, and then take that morphometric data and then go back to the actual molecular ID of them. And we're able to develop techniques where we can sort eggs visually and target the aquaculture of species on what we know that we're raising. So if we want to raise a particular species that are spawning on these exhibits, if we want to do R&D on a species to see how good they are, how easy maybe they are to raise, or if there's some technology that needs to be developed, we can target them without setting up special broodstock tanks and going through all that effort to try to take them off exhibit. We can just use these exhibits really as an innovation hub for testing out a lot of new finfish species that people have not had access to. And there's not a number of species that are spawning on exhibits across the country at public aquariums that have never been accessed by aquaculture, and we can look at families or genus level information about larval rearing. And the last thing I want to talk to you about in the last couple of minutes I have here is some work we have done with a biotechnology company at Roger Williams, and that is Larry Feinberg and Knit Bio. So Larry came to us. I was actually at the New England Aquarium. I used to spend a couple days a week up there. And when he walked in one day, and he was looking to pilot test an idea that he and a collaborator at Harvard had. And that was they were growing bacteria that consumes the decomposition of leaf material. So it's a methanol consuming bacteria. They had engineered this bacteria to be able to do a lot with it. And they wanted to use it as a protein source to feed fish. But he had very limited resources. It was a new, just a fresh startup. They had a little bit of angel funding. They had fermented their bacteria in his barn. They had settled it out, and I had sent it to a nutrition colleague of mine down in North Carolina that had made some food for us. And we basically were able to get Larry and Knit Bio up and running with some very early pilot scale testing of how does this diet compare to fish meal and other replacement diets. And we were able to get him the initial data that really drove his ability to spool his company up. And he actually has a product now that's on the market. And they're kind of in this area for startups. It's a little bit nervous where they have a functional product, they're in commercialization, and they're kind of waiting to try to build market and build capital and revenue to get out of that valley of that area and startup. But it's a very successful product. And I remember the first time we ran an experiment on this, the data looked so good, I had to kind of hold Larry back a little bit. I said, we only have a few replicates here. I know this data looks exceptionally good, but we probably should follow it up to make sure it's this good before you tell the world that this is a great fish meal replacement. So we did all this testing with undergrads at Roger Williams, and we basically took their process and we take their bacteria, and then we were feeding this, and we demonstrated that it's very effective in feed. And so they're very interested in, can this fermented microbial protein or small or single cell protein benefit fish health? Can it help with sustainable aquaculture and by reducing fish meal and soy dependence in aquaculture? So protein is a huge limiting factor to the growth of aquaculture. As we're growing the production of food, we're going to run out of protein source that's available. And so we need alternative proteins. And the data that came back from the initial studies that we've done, we've published this work. The microbial data in their gut was really striking. Usually when you take something other than fish meal, soy, or some other protein concentrate or protein meal, you are going to get some type of different microbial community in the fish's guts. And we actually didn't see that with Knit Bio's products. We saw really no difference in microbial community in their gut, which was really amazing. And further work that Larry has done with commercial farms, on farms in commercial aquaculture settings, has demonstrated really convincing evidence that they're improving survival in stressful events, reducing disease susceptibility, and that probably has to do with the fact that there is some benefit to this bacteria and some of the products in that bacterial meal to fish's gut microbiome and their overall immune health. And it's a very exciting pathway. People are getting really excited about single cell protein. And this is one product. And we really are really proud that we were able to help Knit Bio get scaled up and do that initial work. And so, to stay on time, I wanted to say thank you, and I'm happy to take a few questions if people have them, but I really appreciate the opportunity to talk to you about our program today. Much of my work has been funded by our collaboration with the New England Aquarium, but we also have funding from the Association of Zoos and Aquariums, SeaWorld's Busch Gardens Conservation Fund, with an organization called Rising Tides. And then the Institute of Museum and Library Services is a federal government agency that funds aquariums, and they funded a lot of training workshops for us to build systems and train aquariums how to raise fish. NOAA has supported my work for some time, and I'm supported by the William H. Donner Foundation.
Thank you so much, Dr. Ron, for your talk. And if there's any questions, please type them on the chat, and I'll try to get one or two in for the time. In the meantime, Andy, can you release, please, the screenshots, the screen share, and we don't have any question at this time. Maybe we'll have later. So our next speaker, Dr. Eitan Salmon, who is a researcher at the Israel National Center for Mariculture at the Israel Oceanographic and Limnological Research Center. Dr. Salmon is focused on biotechnology and mariculture application of microalgae and seaweeds. We will hear about the use of algae with antimicrobial activity to promote survival rate and growth of fish fingerlings. Dr. Salmon, please share your screen.
Thank you, Galit. Thank you for inviting me to show a little bit of my research. I should say it's a joint research, of course, with Galit, who used to work here with us. And also would like to thank the previous speaker, Dr. Andrew, because I'm also going to speak a little bit about fish feed, and especially the use of algae with some antimicrobial properties, and how we can help fish survive better against outbreak of bacterial disease. So just we're limited on time, so really just a very short introduction. I kind of sure that most of the people here in the audience are well aware of the things I'm going to say, but I'm going to say them anyway, just briefly. So, of course, with a rising human population, we need to find alternative food sources, especially protein. And of course, mariculture is one of the key factors for it. So the fish mariculture practices are constantly growing. Like I said, mariculture is important, and we need to grow more fish, but of course it comes with the price that the dense fish culture are more susceptible to disease outbreak and rapid spread. And unfortunately, we can't always use antibacterial agents, commercial antibiotics, either because they don't work anymore, but a lot of time because of course, government control, which is very important, of course, because we don't want to contaminate the oceans with too much antibiotics. So there's a need to find new alternatives or new drugs, if you will, and marine microalgae may supply such alternatives. So the microalgae, of course, are unicellular or colony-forming photosynthesizing eukaryotes. Mostly, of course, you have the cyanobacteria, which can also sometimes be considered as algae, depends who you ask. They roughly divided into really multiple taxonomic groups, and the important thing is that each taxonomic group has their own morphological and more importantly, in our sense, is biochemical characteristics. So if you see different colors, different activities, it's because they can produce different substances. Many species are relatively easy to grow on seawater-based growth media. And some microalgae are already serve as maricultural purposes. Some of them, of course, are a product per se, like you can buy chlorella powder, for example, or chlorella pellets and things like that, usually as a food additives. But of course, they also serve as part of the live food chain, which is really a hallmark of the marine aquaculture, and they're required for larval rearing. Some microalgae species have been demonstrated to contain antimicrobial agents, and most of the work that's published today is something like that. The researchers grew some kind of algae, they did an extraction, and then tested the effect of the extract against a certain bacteria or several bacteria, mostly on agar plate assays, and that's it. And we wanted to take this a few steps forward and to test it more deeply to see how far away can we still have the antibacterial effect. So we started with some microalgae species, 2 local algae species that were isolated here in Eilat. And I'm going to call them algae 1 and algae 2. But this is just because we're not sure yet what are the actual species are. We are currently working to find, we only find rough bioinformatic resemblance to other species. So we're really trying to figure out the actual species. They're probably new species, local here to Eilat. We grew them, and the extracts were able to inhibit the growth of gram-negative bacteria, the Vibrio paralyticus, and gram-positive bacteria, which is Streptococcus agalactiae. Both of them are disease-causing bacterial agents in fish. So the process is relatively simple at this stage. We grow the microalgae, a few hundred liters. We concentrate them with a centrifuge and oven dry these algae. We grind them into a powder and do some acetone extraction. This is simple solid phase extraction. And the extracts themselves, we do the antibacterial assays, and we found some effects. I will perhaps show larger version here. You can see the paper disc with the extract, and then you have some rings of the inhibition zones of the growth of the seaweed, of the bacteria. And it's interesting to see that you have actually, you guys can see, you have 2 rings, concentric rings. One of them is a clear ring immediately surrounding the inhibition zones. We suspect we have several materials, not just one. We have these 2 algae, and the next step was to take the algae powder and produce 5 feed species for feeding and bacterial inoculation test. We have a control. That's the regular food we give our sea bream, that's the fish we work with. And the other diets, we added either 5% of algae powder replacing the diet, and or the acetone extract that we did. We actually did the acetone extraction, then completely dehydrated it, and then immersed it, solubilized it in the fish oil content of the diets. And we had 5 diets: algae 1, algae 2, extract, and dry powder. We grew the fish, the sea bream fingerlings, about 15 grams each. We gave them these diets for 30 days. Of course, everything is done in triplicates at the very least. And at day 30, we did some subcutaneous inoculation with LD50 dosage of Strep agalactiae. This was done with injection. This was because of technical issues. We couldn't just spread a lot of bacteria into the water. And to keep the use of bacteria limited, we have to bypass the skin, the fish skin, which of course is very important to its defense against. Can you hear me? Excuse me.
Yes, we can.
Okay. Sorry. Okay.
Please mute your microphones. Other people, please mute your microphones. Thank you.
Yes. So we injected the bacteria directly to the muscle, and then followed the disease development for 10 days. We took out the dead fish. Of course, we counted them. And the survivors, we sampled the serum. We sampled, also did some microscope work. And I will just show you the results. So the main result for this experiment is this one, that addition of the algae 1, either dried or extract, to the feed resulted in lower mortality rates. You can see the mortality rates and the different diets was down from around 57, 58, down to 48 or 50. So that's almost, in some cases, 9 to 8% drop in mortality, which of course means increased survival. For algae number 2, we found that when we added just the dry algae, we didn't get this effect. But when we added the extract, we did have some effect. So we wondered whether it's because of some case of immune response of the system to the food. And this is probably not the case. We took out serum and measured the total antibody levels. And for the first 30 days, we just got the food. We didn't see any significant rise in total antibody levels. But after inoculation with the bacteria, the amount, of course, doubled. So this led us to believe that probably the effect is coming from the food, from antibacterial activity in the feed itself. Of course, we had to check whether or not it's problematic to the fish, the feed themselves. So the diets had no significant effect on the fish growth rate. If anything, it actually kind of improved the growth rates of the fish, at least algae 1, which of course is our star candidate right now. And they had no significant effect on the nutritional values of the fish compared to the control. We took the fish, we measured the content of the ash, lipid, and protein. We didn't see any significant change. All this tells us that we have these two local microalgae that are capable of inhibiting growth of several types of bacteria. We haven't identified the substances yet, but we do know that, and this is important, we know that they can withstand the oven drying process and the extraction process in acetone. Both of them are in 60 degrees Celsius. And they still retain activity even after the pelleting process, which uses steam to produce. I was very worried about this part, that we're going to lose activity. The dry algae powder extract can replace up to at least 5% of the total feed, with no effect on the growth of the fish. And the antibacterial activity, this is really was kind of surprising a bit for me, that we still had this 10% increase in fish survival, even after we actually injected the bacteria into the fish themselves. We expect that we will get better results if we have less invasive inoculation methods. I mean, when the fish, usually fish in nature, they will be inoculated by bacteria, usually from the water body itself, and not by a sharp object, sometimes yes, of course, but in many cases, the spread of the bacteria comes through the water, and we expect to have even better results and survival of fish when inoculated in a more or less severe manners. So just to conclude, the addition of certain microalgae to the fish feed can improve the resistance to bacterial disease outbreak. In our case of algae 1, the simple addition of just the dry powder gave good results. And this, of course, saves us the costly and polluting extraction steps of the acetone, and we don't need it, we just add it directly. Of course, we need some further work to explore the possibility of increasing the microalgae quota in the feed, because the algae themselves also have proteins, they have nutritional values in themselves, so they can perhaps replace, I don't know if the entire fish meal, for this we can use Andrew's friend's protein, but they can use, maybe perhaps we can exchange more of the feed and save something on it and get a better fish and healthier fish. Of course, there's a need to expand the experiment to other bacteria, other fish species, and to identify the active ingredients. We just say that currently we are looking at implications for human health with the same bacteria, and we have some promising results. So I would just like to thank, first of all, the people at my institute. Really, a lot of people were involved because this is really a multidisciplinary research. We had Galit and from fish pathology, and Ama, she will also talk today, with the algae growth, and Shaital and others with the chemistry and nutrition department. And really, most of our institute took some part of this experiment, and of course the funding agency, the Ministry of Agriculture and Rural Development, and the JCA in Israel. And thank you for listening. I'll be happy to take some questions if we have 1 minute. I see one question. Do you have data on ease of upscaling algae? Well, these are we didn't try to do upscaling, but we did work with large quantities already to begin with. So we grew a few hundred of liters each time. And I expect that to upscale it even further shouldn't really be too much problem, especially for the algae one species, which we found is easier to grow. It doesn't sink to the bottom so much, like sometimes happen with microalgae, and easy to count, easy to harvest, relatively easy to dry, even in large quantities. So I don't expect it's going to be too much of a problem. Thanks.
Thank you, Dr. Solomon. Our next presenter is Dr. Robert Holmberg, and Dr. Holmberg is an assistant professor of biology and shellfish aquaculture and a hatchery specialist. He's also a member of our SEED at Roger Williams University, and Dr. Holmberg will be talking about improving the resilience of hatchery-reared blue mussels to ocean acidification and with diet and seawater buffering. Dr. Holmberg, please.
Thank you, Galit. Can everyone hear me and see my screen?
Yes.
All right, great. Once again, I'm Rob Holmberg. I'm co-running the research program in the shellfish hatchery at Roger Williams. And today I'm presenting on this project that I initiated as a postdoc at my previous organization, the Down East Institute in Maine. And like Galit said, it's all about improving resilience of blue mussel larvae to ocean acidification stress in a hatchery scenario. All right, so the blue mussel industry is worth about $3.6 million in Maine. And the way it used to work is that mussel farmers would just hang ropes in the ocean at the right time of year, and wild blue mussel seed would settle on the ropes, and then they'd grow them out to harvestable size and sell them. But in recent years, wild blue mussel seed stocks have sharply declined, and mussel farmers are relying more and more on hatcheries to produce the seed for them. So the way DEI does it is they spawn blue mussels in their hatchery and then settle them onto ropes in the hatchery, pass the ropes onto the farmers, and then the farmers put them in the ocean and grow them out and share a cut of their profit with DEI. But why are blue mussels declining in the wild? Until now, it's likely due to predation by the invasive European green crab. So ocean warming is increasing the range, metabolism, and growing season of the green crab. So they're eating more mussels in more places and later in the season. This is data showing that soft-shell clam recruitment decreases as green crab mass increases. And it's likely the same thing is happening with blue mussels. It's pretty rare now to find wild blue mussels in any abundance on the Maine coast, except on floating docks and buoys where the crabs can't get them. But as we move into the future, it's likely that ocean acidification is going to be a second stressor on wild blue mussels. We know that experimental OA reduces larval blue mussel hatching, survival, shell thickness, and tissue weight. So there's reason enough to think ahead about how we might mitigate this in the hatchery. So DEI hired me as a postdoc in 2019 to build an ocean acidification lab for studying OA and warming impacts on larval bivalves. It consists of 32 experimental conical aquaria. And using the system, it's possible to manipulate seawater pH and temperature fully independently in every one of the tanks. So it's really good for setting up full factorial ocean acidification and warming experiments. And I've used it to study the effects of those things on larval softshell clams, larval surf clams, larval lobsters, and now larval blue mussels. So how do hatcheries normally adjust seawater pH? Until now, most hatcheries have been using seawater buffering. So they add a buffering agent like soda ash, aka sodium carbonate, to bring the pH back up. But not every hatchery has the equipment or the expertise necessary to do that. So I proposed using alternative live microalgae diets to feed the blue mussels. hopefully in a way that makes them more resilient to climate stress. DEI cultures as many as 13 different species of microalgae. So I had a lot to draw from. And I set up an experiment consisting of 2 seawater pH treatment levels, 7.8 and 7.3, which represent present day and year 2100 coastal Gulf of Maine pH conditions. I chose 4 different diet treatment levels. We tend to pair a flagellate and a diatom. So the standard blue mussels diet used at DEI is a combination of T-ISO and T-SUDO. So that was one treatment. I chose another treatment, which I call the modified industry standard diet, which is feeding them T-ISO and T-SUDO as veligers, and then Rhodo and Weiss as petty veligers when they're big enough to eat that. And then I chose another treatment consisting of T-ISO and Chagra, which is a high DHA treatment. And finally, mono and Chagra, which is both a high EPA and a high protein treatment. So then I reared larval blue mussels between fertilization and about a week post-settlement before sampling them, and then measured them for survival and morphometrics. And I actually settled them under experimental conditions in my experimental tanks onto short sections of rope. So this experiment also had a field deployment phase. So I worked with 2 blue mussel farmers in Maine, Blue Hill Bay mussels and Bangs Island mussels, which are located on very different parts of the Maine coast, one down east and one in southern Maine. So we took all of the short sections of rope that I produced during the experiment and then attached them to longer ropes and then suspended them on these blue mussel rafts at the farm's lease sites. And then I let them grow out for between 6 and 8 months. So this is a photo of the mussels after about 2 months. I actually initially deployed them in a secluded salt pond area before moving them into the open ocean at the farms. This is what they looked like after about 2 months in the salt pond. And then along with the mussels, I also deployed some sonde equipment to monitor water conditions in the ocean. And I'm monitoring seawater pH, PCO2, salinity, temperature, and chlorophyll. So this is an ongoing project, and I'm not ready to share any preliminary results yet. But the experiment I just described is the first of 3. Experiment number 2 consists of 3 different variables, pH, diet, and seawater buffering, to test whether diet could be more effective than buffering, or if there's some interaction between diet and seawater buffering. The lab trial for that experiment is complete, and the field deployment, along with the deployment from the first experiment, is currently underway. And then finally, once those are done, I'm planning on doing a 3rd experiment, which consists of pH, diet, and buffering, but scaled up to commercial levels using the large larval tanks in DEI's hatchery. And I'm planning on completing that trial this summer and starting the field deployment this summer. So I'd just like to thank all of my collaborators at DEI, as well as my new colleagues at Roger Williams University. I'd also like to thank Sea Grant, who funded this project, and I'll take any questions.
Thank you. We're waiting for some questions. And in the meantime, if you can share your screen, and we will be able to move on to the next speaker. If you have any questions, please write them on the chat.
Is it possible to ask a question here?
Yeah,
Yeah, so first of all, thank you for the informative information. Could you give the full Latin names of the microalgae species you used? Because I wasn't familiar of all the names that you mentioned.
Let me get back to you in a minute and put the names in the
Okay,
chat.
no worries.
Yeah.
We can contact after. Just another additional question. Did you run trials regarding a depuration process as well? Is it something which is practiced in mussel cultivation, same as oysters and other bivalves which are marketed for human consumption?
I haven't run any trials like that, but I think that would be a really interesting project.
Okay, thank you. So I'll approach you after this webinar, and we'll see how we can find maybe mutual interest. Thanks.
Great.
Thank you very much.
this is exactly why we are here for. So thank you so much. And I'll introduce our next presenter. I want to introduce a dear colleague of mine, Dr. Naama Segal. Dr. Segal is a senior scientist in the Israeli Oceanographic and Limnological Research Institute, working at the National Center for Mariculture in Eilat. Dr. Segal will be talking about identification of algae-derived compounds for the use in the alternative protein and the cultivated seafood industries. Dr. Segal, floor is yours.
Okay. Thank you for that. First, I would start by thanking Galit for letting me present my work here at this conference and giving me the opportunity to present what we do. Eitan spoke before a bit about some aspect of our work, and I wanted to present to you our institute as well. You can see we are in Eilat, right at the border to Jordan, very close to the sea. And in our institute, we are focusing on many different aspects of mariculture. And this research is kind of different because it's taking the use of algae to industries that are more related to cultivated meat and seafood industries and alternative protein, which are not the focus of mariculture, but also develop along it, and algae can support those industries in different ways. I'll start by introducing my unit. So as you can see in my unit at the NCM, we can grow different kind of algae. Some are industrial and some are locally isolated. This is something that we are always working on and always and all the time trying to isolate more species from the fish ponds, from the Red Sea directly, and try to see how we can manipulate them and use them for different industries and uses. We have an indoor growth room where we can grow the algae from agar plates to large plastic PBRs, where we can hold about 3,000 liters just in the room, and we can control the light. We also have an outside part of the unit where we can grow the algae in raceway ponds, and we are developing more compartments where we can grow the algae in more systems where we control the operations more, but we can also take those large amount of algae and use a centrifuge to concentrate them, and then we can develop them to we can dry them into a powder and extract them and do many things with them. And this ability gives us the fact that we can use our research not just in the lab, but also to try and utilize it in much larger scale, like the fish experiment Eitan described. And we wanted to utilize those different algae that we have into different aspects, and not just to use as a feed, or when we talked about food additives, we usually talk about the protein of the algae, but we wanted to look on other substances the algae has within them. And you can see our diatoms collections where we can extract their pigments, and we can see the fucoxanthin and other pigments they have within them at the growth conditions. And we can try and utilize those pigments into food additives and color additives into food, and the alternative protein industry like salmon mimicking alternative proteins for utilization of this. And also from Gracilaria, you have the phycobellins that also being used in this industry. And you can, by the extraction method and pH, control the pigment itself and see how it can be utilized in the industry, and it's used as the color they need for the product. Also, the algae are, whoever encountered dried algae or spirulina or in the past know they have a very strong smell. And while sometimes it can be a disadvantage, at this industry it can be an advantage. It can help give alternative protein which come from either peas or other materials, a sense of fishy smell, as marine algae has a very strong odor to them. And we are trying to, from our algae collection, we are trying to identify and see which are the volatiles and the other active compounds within those algae that can be further utilized as food additives and flavor additives in this industry. So we are examining, this is a collaboration with another group from the north of Israel, with Dr. Suleiman Khatib, and we are analyzing this algae and identifying the active compounds, and we compare it to databases and other fish and seafood, so we can see which algae mimics the best of which algae mimics best the seafood that we want to mimic. Again, so other than the proteins and the color additives and flavor additives that we can find in algae, in macroalgae, here you can see Gracilaria in the picture. We can also find different polysaccharides that can be further utilized as scaffolds to grow on them the fish cells or the seafood cultured meat, and also in regular cultured meat, and to try to integrate those scaffolds and polysaccharides in this industry for texture and also for actual scaffold to the cells to grow on. So this is the this is one last thing. Another aspect we are looking on in this algae we are examining is to see their materials, and to see if we can replace different substances in the serums that are used to grow cells in cultured meat industry and cultured seafood industry, and to replace the serums that are being used today to an animal-free growth media. And we are examining different algae to see if they are capable and have the ability to contribute to this aspect of this industry as well. And this will enable us to have or to enlarge the market of microalgae and macroalgae to other industries as well, and not just the use of them as just for their protein or just the use of them for their feed. So this is another aspect of what we are doing in my lab and in the institute. And I want to thank my lab members and my project collaborators, Eitan and Suleiman, for doing this project with me. This is a very exciting project that we are in the middle of it. I'm not presenting any data because we are still, it's a really ongoing work, and I'll be happy to talk to anyone this is interesting for you.
Melody Walsh, sorry. Please present your screen, a PhD student at Professor Amir Sagi Laboratory.
Okay, so hello everyone. My name is Melody Walsh. I'm a PhD student from the Amir Sagi Lab at the Ben Gurion University. And today I'm going to talk about sustainable all-female grow-out in the freshwater-prone Macrobacillus rosenbergi. Okay, so Macrobacillus rosenbergi is a decapod crustacea native to the Southeast Asia, and it's cultured almost all over the world. And this prawn is highly environmentally adaptive, and its adaptivity places it as a species with highly invasive potential, especially through aquaculture escapees, as with reports of invasion in South America and Africa. And the typical distribution of adult population, of mixed population, includes females and three morphotypes of male. The small male, you can see it here in the picture, the orange claw male, and the dominant large blue claw male. So this is how the typical mixed population looks like. Then why monosex? So the use of monosex population is very common in animal husbandry, and it's favorable also in aquaculture mass production with better performance compared to mixed population. Now, these animals are non-breeding monosex animals, so they cannot establish themselves in the environment. So it reduces the risk of invasion cases as a result of aquaculture escapees. And another interesting environmental management of this monosex population of prawn is the use of this prawn as a biocontrol agent against pest freshwater snails. So you remember that this distribution I showed before, and we can see there is like a variation in the size in the population, but there is one group that its size is uniform, the females. So why females? The females, they grow uniformly, they're less aggressive than males, and they can be stocked in high densities. So what controls the sexual differentiation in prawns? The process starts with the genotypic sex determination, the WZ or ZZ chromosomes. Then we have embryonic stage, larvae stage, and after metamorphosis, the post-larva stage. And then at the early post-larva stage, there is a sexual switch. Then animal with ZZ chromosome, this is on. Then AG, androgenic gland, is developed and secretes hormone called IAG, the insulin-like androgenic gland hormone. Then we get eventually the phenotype of the mature male. Now, animal with a WZ chromosome, the switch is off, so there is no AG, no androgenic gland, therefore no IAG secretion, and we get the mature female. So to this sexual switch, we call the IAG switch. In normal case, breeding of a WZ female and ZZ male produce 50% WZ females and 50% ZZ males. So what do we need in order to get an all WZ female? Super female, WW female. When breeding with the normal ZZ male, the yield will be 100% of WZ female. So the biotechnology of the WW female production starts with AG cell suspension from a male donor. These suspended AG cells transplanted into a normal WZ female, and it caused a full sex reversal into WZ neo-male. This WZ male, when crossing it with normal WZ female, 25% of the offspring will be WW female. Now, these females can be manipulated again. The manipulation, the transplantation happens at the early stage of the PL of post-RV, as I showed in the IG switch slide. And then this WW female will become a WW neo-male. Then breeding with WW female, we get the 100% WW female offspring. This work was done by Dr. Tom Levy, a former student at the Samuel Segui lab. So now we have consecutive generations of WW females without the Z chromosomes, with no abnormalities compared to the WZ normal female. So these females can do breeding with any normal ZZ male in order to produce all female population. So we can say that the WW population can be utilized as a brood stock for commercial all female. Okay, here you can see some results from commercial scale all female grow out experiment. You can see here the very nice uniformity of the size of the females, and notice to the very impressive densities, 12, 16, and even 20 prone per square meter. Just that you know that the traditional mixed sex culture is that densities are about 4, 5, 6 prones per square meter. So this is very high density culture. And the most important, one of the important aspects, I think, is that it's also worthy. The total yield of all female culture at the density of 16 per square meter is about 3,000 kg per hectare, compared to the traditional mixed sex culture of 6 prone per square meter. It's about 1,200 kg per hectare. So we can say that this all-female farming strategy is superior to the traditional mixed sex culture in terms of density, stocking density, of the size uniformity, and also at the total yield at the end of the harvesting. Thank you very much. I would like to thank Professor Amir Segui and the Amir Segui lab, the Enzutic team, and I'll take questions now. Thank you.
Thank you so much, Melody. Sorry for mispronouncing your name before.
Okay.
So thank you very much. It was very interesting. If anybody has questions for Melody, please, this is the time.
Well, Galit, I have a question. Sorry.
Yeah.
Yes.
Yes, Dr.
Yeah,
Ryan.
so Macrobranchium have long been challenging because of getting an all-female population, right? I'm really surprised to see that you can use the same chromosome system or similar methods that we used for genetically male tilapia in this system. It seems like it has so many commercial applications to solve the problem of aggression and the reduced density. Where are you at in the process of commercializing these broodstock? I mean, you can currently buy genetically male tilapia, right? Because they're a valuable broodstock resource. So where are you at with that?
The culturing of the Macrobranchium all female, it's already in the commercial scale.
No, it is. Okay, so they're already available in the marketplace.
Yes. And do you
Well,
think
that's
yeah.
No, that's great. Colleagues of mine, when I was a postdoc, were heavily invested into Macrobranchian culture, and the vein of their existence really was the problem. And there had been a lot of effort, Luda Brahma, a lot of people have spent a lot of time trying to think about how to do this. It's really nice to see that this has been accomplished.
Yes, the aggressiveness is part of the less aggressiveness, yes, because the male are with the aggressive behavior.
Sure.
So the female, you can grow them all female, and then you just take it off from the equation, and you get very uniform size, and they get even bigger because they're a culture without males, so that they almost don't stop early as
right. It's like tilapia but reversed. Yeah, it's excellent. Excellent.
So thank you very much.
Has a very high sexuality. Sorry, no, it's okay. Sexual plasticity of Macrobranchium, it's amazing.
So thank you very much, Melody. It was really interesting, and we will continue with our next speaker. And I'm happy to present Mr. Benjamin Getch, the aquaculture coordinator at the Rhode Island Management Council. We will hear about marine aquaculture in Rhode Island, overview and permitting processes. So Benjamin, please, if you can share your screen.
So my name is Ben Getch. I'm the Rhode Island Aquaculture Coordinator, and I work at the Coastal Resource Management Council. A little bit about CRMC, my agency. We are responsible for the management of the coastal zone and submerged waters of the state. So generally, if you're going to build a house on the shore or any other kind of development, a dock, a marina, you need a permit from us. We've got other responsibilities under federal consistency with the federal government, and we see that play out most recently with the offshore wind development in some of our offshore waters. But we are also the primary agency for oversight of the aquaculture program in the state. And we also engage in special area management plans, which are kind of ecosystem-based strategies to tackle different challenges, and we've done them for various different things, geographically or themed, with ocean change, sea level rise, and watershed management. But we've most recently done one for our bay, Narragansett Bay, and that was primarily to accommodate this renewable energy cable corridor, which would be plugging into the offshore wind farms, but that also provided an opportunity for us to explore aquaculture regulation and any new changes we might want to initiate. I'll touch on that a little bit later. But just to illustrate our jurisdiction for purposes of federal consistency, so this would be waters outside of the state's jurisdiction. It'd be federal waters, but through a mechanism in the Coastal Zone Management Plan, we are able to project some limited jurisdiction out into the federal water, and that is to ensure that any federal action out there would be consistent with our Coastal Resource Management Plan as approved by NOAA. And like I said, this play has been playing out with regards to offshore wind development, but it also applies to aquaculture activities, particularly in this 2011 GLD, which is our geographic location descriptor. So within that dotted line around Block Island and Long Island and Rhode Island Sound, we would have some review over aquaculture applications in the offshore environment. We currently have none, but I just put that there for your all reference. But to go back in time and just to do a little history, I always like to go to the beginning. Oyster aquaculture is really what we have in the state, and it is kind of a legacy industry that dates back, well over 100 years. The industry really peaked in about 1910. Here you can see almost a quarter of the bay was leased out, and this was bottom planting, primarily with seed oysters that were sourced from other regions, Long Island Sound, the Chesapeake Bay. But it did result in something of a mill town kind of labor system, which was not looked upon fondly. And the industry did decline really with the hurricane of 1938, putting the kind of death knell on the old leases. And the last oyster lease historical lease was given up in 1952. And this had a lot to do also with World War II, a lot of GIs coming back and wanting to abolish these kind of labor systems. And this was kind of the rise of what we call the free and common fishery, which are the commercial shell fishermen that harvest from the wild stocks. But anyways, we had a lot of pollution issues that took a while to get our hands around. Changes in our coastal uses, developments, a lot of tourism, and this kind of not so fond memory of what the aquaculture industry was. But that started to change in the 1920s. Luther Blount, named after the Blount, the hatchery at Roger Williams is named after him. He got one of the first experimental permits on Prudence Island to try some oyster culture. There was also a mussel farm, the Blue Gold Mussel Farm, which still exists in some form, though it's a different company. But it was really in the 80s that there was a moratorium on aquaculture, and there was all kinds of issues with hearings between various competing agencies had their hands in this process. Even though it was first laid out, CRMC was only created in the late, early 70s. So we had our regulations, but it was still difficult. It really was a test of the process came in the 1988 with the SPATCO application. That's Bob Rowe. If any of you guys know Bob, he's the president of the East Coast Shellfish Growers Association, but really the grandfather kind of modern oyster shellfish aquaculture in Rhode Island and even the region. But after that contentious application, there was a couple of legislation actions that really made my agency, Coastal Resource Management Council, the lead agency. It also was able to secure funding from Senator Jack Reed, who continues to support aquaculture at Roger Williams as well. But that also created my position, the aquaculture coordinator, and kind of gave the mandate to the agency to develop aquaculture in a responsible way. So this is what kind of came out of that session. This is our Aquaculture Act, and you can see, the process of aquaculture has been determined to be a proper and effective method to cultivate plant and animal life. Now, that was important actually to get out there and state, but it also says that it has to be conducted in the best manner of the public interest, which is always hard to discern. But we are given some guidance in that we have to give particular effect to the free and common fishery, which I said again is those wild harvest shell fishermen who are kind of our cowboys here in Rhode Island. They're kind of a charismatic megafauna of our bay, so to speak, but also the compatibility of the aquaculture with the environment, obviously. So that is our guiding enabling legislation. We also, interestingly, have jurisdiction over marine, not just marine aquaculture, but freshwater and land-based operations. And that also includes ornamental fish production. And this is actually one of the places where CRMC's jurisdiction extends well beyond the coastal zone. It's really one of the only areas. But we do work with our sister agency, the Department of Environmental Management, on a lot of these reviews and the application process. Also, the Marine Fishery Council is an important contributor to this process. And we also have a biosecurity board, which faculty at Roger Williams have contributed to. And that board will have to approve any species that are non-native that were intended to be grown here, even in land-based operations. And they're going to want a review of operational plans and security protocols. But we also work with USDA and others on crop health and disease surveillance. So we recognize that aquaculture is a viable means for supplementing, marine fish and shellfish, and where it can be accommodated over those other uses. And that really is the rub here. If there is a barrier to expanding the aquaculture industry, it is this issue of accommodation. It is becoming more complex and contentious as the years have gone on. And now we see many applications being contested and delayed many years, ending up even in the court system after the CRMC's approval or denial. But we do try to be proactive and work with a lot of stakeholders. We've got a couple examples of that I'll touch on. But we work with all the universities, such as Roger Williams and URI, and also the industry, supporting research and things like that. We have a lot of regulations and restrictions that have kind of evolved over the years, and we're all trying to maintain this balance between the public trust waters, between the public rights and aquaculture development. It's always a balancing act. But as we have grown the industry quite a bit since the early 2000s, and we've gone through a couple different working group policy planning iterations. The Shellfish Management Plan was one. CRMC Working Group on Aquaculture Regulations has met a few times over the years. And we also work with oyster restoration. So we have a different flavors of permits. Water-based aquaculture, your marine tidal aquaculture, it's going to be an oyster farm, 9.9 times out of 10. Commercial viability gives you a limited permit for just 3 years and allows you to test the commercial viability of techniques or species that may not be yet proven, or to test the viability of a particular area with regards to those. But we also have research educational permits that are, again, limited to 3 years, and they're also limited to about 1,000 square feet. You can apply for more, but it might be more of a permit process. But we also allow for experimental and recreational permits as well. A recreational permit is you have to have a dock to do that, and your dock has to be in approved waters for the consumption of shellfish. But you're allowed to grow a small amount of oysters. It's a fun program. But anyways, our applications, require, various things, the operational plan being one of the most important, next to, site plans, diagrams, things like that. But here's a look at the industry over time. This is our dollar value of aquaculture, of all aquaculture products. And you can see a bit of an exponential curve. It's starting to curve up quite a bit. Around 2012, 13, 14, we see a big jump. And, we got a lot of people who started using floating gear at this time, and expanding into areas and improving operations through adopting new techniques and technologies. Of course, there's the COVID dip, about a $2 million deficit in 2020, but has sprung back as strong as ever. And likewise, our acreage and number of farms is listed here. We're at about, this is actually a bit outdated, we're about 374 acres right now and 84 sites. But just to highlight a few key moments in time, this was the Aquaculture Act that created CRMC as the lead agency and designated an aquaculture coordinator to help the permit process. This is our first working group in 2007. Which really focused on the ponds are salt ponds, which are the shallow lagoons in southern Rhode Island. And that came up with a 5% limitation on aquaculture in a water body. So based on the area of the water body, 5% of that was deemed to be allowable to devote to aquaculture. And now we're working now with our new kind of base AMP working group. We did that over the last few years. And really what came out of that was kind of a more engagement with stakeholders in the process. We didn't really make any regulation changes, but we made some notification changes and things like that. But where is aquaculture currently? Like I said, it's all oysters. We have like maybe 2 kelp farms, sugar kelp, very little quahog hard clam production, a little bit of bay scallop, but this is 99.9% oysters. And as you can see, the real important places by acreage, but also by production are Ninigret Pond, just a small pond here, Point Judith Pond, historically, it's always been an important center. And more recently, the Rome Point, which is in the West Passage in Dutch Island Harbor, which is off Jamestown, have become very important areas and support some floating oyster gear operations, which do quite well. So that 5% limitation, as I mentioned, again, this is a little outdated. We're at about 370 acres, but these percentages haven't really changed. There's a 5% cap, so Point Judith Pond is pretty much at 5%, and the rest of the ponds have some room left, but are constrained based on other reasons, whether it be natural resource conflicts like submerged aquatic vegetation, user conflicts, or other management programs like spawning sanctuaries or shellfish management areas. But these are generally the grow-out strategies we see employed in the state. Now I have an X on land-based and intertidal because we simply don't have any land-based commercial operations in Rhode Island as of yet. We have some research facilities, obviously, such as at Roger Williams and URI, but we don't really allow intertidal aquaculture in the intertidal flat areas. We avoid those, and that has something to do with our kind of constitution here in Rhode Island that all citizens have privileges of the shore that can be exercised in that intertidal area. So we really don't encourage that, but we have subtidal, floating, suspended, and on-bottom type operations. So, on-bottom culture, I'm sure you're all kind of familiar with what it looks like. We have some trays, some racks and bags, nothing really to see on the surface unless it's a really shallow area and you get in a plane. But that's pretty typical of what we see, particularly in the salt ponds. The harvest methods are, pulling up the cages like a lobster pot a lot of times, or just working in the water in waders. You can see some various techniques. These are all Rhode Island images. And we also suspended culture, and these are using kind of they call it stacking trays, suspended on long lines. We have 1 or 2 operations that utilize this method, but you can see the boats are a lot larger. Infrastructure is a bit more. Those same companies also often grow mussels and have historically grown mussels and transition into oyster culture using the same kind of infrastructure, long line systems. In sugar kelp, as I said, an emerging industry. We have 2 or 3 folks a year that grow it. I think we had about 14,000 pounds last year. But the real game changer has been floating gear for oyster cultivation. This is a typical diagram of what this looks like. It allows you to flip the gear upside down to dry it, desiccate any biofouling, also improves shell strength and condition of the animal overall. This is a much more marketable product, and mortality is dramatically lower. So here's some images of different types of floating gear that are being used currently in Rhode Island. But so during our most recent policy, special area management plan for Narragansett Bay, one of the things we did was we first wanted to find places where we could direct aquaculture for development. That proved to be incredibly difficult for a number of reasons, mostly because there's just so many different types of aquaculture. But what we were able to do is create a constraints map, a map that says these are the areas you can't go for whatever reason. And we have that now as a tool on our website. And we've also, as part of these changes, like I said, we have new notification procedures to the local property owners, towns. We've always worked with our towns, but we always, we work closely with these local authorities. They don't have any jurisdiction when it comes to aquaculture permitting, but we do work with them as stakeholders and partners in the process. So this is kind of what the permitting process looks like now. It's fairly involved. It can take a year or more to get a permit these days. If you're really lucky, it might be less. It might be maybe 9 months or 6 months, but it really depends on what you run into as far as public opposition, because that will require public hearings. But we do have 30-day public notices, DEM, Army Corps, Historical Preservation, and then our Marine Fisheries Council and Shellfish Advisory Panel all are required to weigh in on these, as well as the public, the general public, are allowed to comment. But we do see a lot more organized opposition, and this is coming from stakeholder groups such as fishing, recreational fishing groups, neighborhood associations. It's become increasingly difficult to navigate this process, just given the kind of current state of society, really, in general. Ever since COVID, things have seemed to be a bit worse, though there's always been some pushback against aquaculture development, though many folks support and appreciate the industry. Of course, they're not the ones who are writing letters, but that's the end of my presentation. A couple resources here for you that I talked about. This is our map viewer, which has some of those constraints and other tools there for notification. The DM Marine Fisheries map is our standard resource reference tool, which will tell you all kinds of other information. But also recently, that just came out, the best management practices for the East Coast shellfish industry came out, and we will be posting those on our website as well and encouraging applicants to explore those as part of their application process. Thank you.
Thank you so much, Ben, for your presentation. Any questions? Okay, so I'm very happy to present our next speaker. So our next speaker is the Business Development Director of Gyro Robotics, Ms. Dee Chatham. Gyro Robotics is a local company which we in Seed have the pleasure of collaborating with in an interesting and innovative project. We will hear about Gyro Robotics' autonomous vehicle in support of aquaculture. And before that, Avi would like to say a one sentence before Dee starts her presentation.
Thank you for letting me say a 1 minute comment. This is a continuation of an effort to connect Rhode Island and Israel. And basically, 2 years ago, we had a competition between the Blue Tech hub in Ashdod Tech, which is for the non-Israelis, the largest port in Israel, for 5th largest city, and innovation hub in Newport, Rhode Island. And basically, Israeli startups presented to Rhode Island judges, and vice versa, the startups from Rhode Island presented to the Israeli judges. So there's no country bias, and the winners received their first prize ticket, but the idea was to connect. The Israeli company was Nak-I. They developed a technology to clean ships as they traveled to reduce energy and environmental hazard, because it would be less time in time. And I'm happy to say that the Rhode Island winners was Gyro Robotics. So as you can see, we're trying to do continue. It's not just a one effort, but trying to be the whole spectrum. So just wanted to give this comment and leave it from there.
Dee, please.
Okay, great. Thank you. Are you seeing my screen?
Yes, we are seeing you and hearing you. Thank you.
Okay, so it's been an amazing morning of immersing with aquaculture researchers and policy people and industry academic partnerships. And so thank you so much for the opportunity to have us here today. It's been great to be working with you so far, Galit, and I'm looking forward to our continuing relationship. So Jaya Robotics was started with the vision to democratize aquatic data collection. And in large part, the way that we do that is to, just wanted to check that my screen is advancing. Yes? Is my screen advancing?
Yes, it is. Yes, it is.
Okay, okay, we got it. Great, perfect. Yeah, so in large part, the way that we're doing that is by providing a very providing access to aquatic data collection with a very easy to use and portable device that we've built. So everything that we really do at Jaya is we have basically simplicity is at the core. Okay, so it's the way we build our business, it's the way we've built our products, and the way our users experience their use with the product. In terms of collecting data and the tools that we provide to help them visualize that data. So what you see here is on the right, you'll see how the JayaBot makes vertical profile dives down to the bottom. So it's deployed into the water, and you can see on the left there, there's what we call a pod, which is a group of JayaBots traversing the surface of the water. So there's surface transit while they're collecting data, and also vertical profile dives right through the entire water column. So let me advance here. Okay, there we go. So we've built the JayaBot in a very modular design, and the idea is thinking that we have one platform and it can handle many applications. We provide it with open sourced software. So many of our users now are people who are taking the basic functionality of the JayaBot and extending it to use in their own research. It's provided with a secure cloud hosted toolkit so that you can set a mission for your JayaBots in the water from wherever you are in the world, from your office or down from the beach or a dock or a boat. And you can launch the JayaBots wherever you want. And you can also be sort of controlling their mission from a remote situation. So there's a very easy to use mission simulator. There's a repository for data. And then there's also a great collection of tools to use to help you visualize the data. This is what we call the basic model of the JayaBot. We call it the JayaBot Hydro. This is commercially available. We have many customers using it already. It is outfitted with sensors to provide data on conductivity, temperature, pressure, bathymetry, such as like what's the bottom type and what's the depth, and then current vectors and wave heights. So it's what some people refer to as a basic CTD machine or vehicle. And it forms really the basis for our whole entire product line. This is the next model that we're developing that's under development now. It's called the JayaBot Echo, and it has hydrophones that are attached, as you can see here, to do some listening underwater. And this is very useful for marine mammal detection and for fisheries, and has really been requested by people in the offshore renewables, offshore wind market, who are interested in what's happening around their wind farms. Our next model under development is the Jayabot Bio, which I think is of most interest potentially to this group. And it is where we've added additional sensors. You can see them on the top here for dissolved oxygen, turbidity, CO2, pH, so that we can tell more about what's going on in the water, and is really doing more water quality testing kinds of applications, detecting algal blooms, and even pollution and oil spills, but we think will be quite useful in the aquaculture and fisheries markets. This is a look at our mission planner, really. So the mission planner is software that comes along with the Jayabot system, and it is super easy to use. We just train people on creating missions within a couple of hours. And so here's an example that you see here, which is a mission that's set up with 5 different Jayabots to go survey a particular area right offshore. So they're launching the Jayabots offshore here, telling each one to go to their starting point, which you see along the bottom, and then what to do when they get there. So if they want them to go down, for example, go down 10 meters, stop and collect data, go down another 10 meters, another 10 meters, go down to the bottom, tell us what the bottom is like. And then finish that, go to the next spot. Oops, sorry. Go to the next spot and do the same thing, So this, I mean, one of the cool things about this Jayabot system is that we're keeping people safe, we're keeping them out of boats. You can really do this from the shore, and the whole launch, survey, recovery process here would be potentially about 15 minutes to get your Jayabots back. And what they've done right here is really surveyed 100 meter by 100 meter site. So we think that this could be really useful for some of the site survey and the permitting requirements that we just heard about from our speaker from the CRNC, and then also in ongoing monitoring around aquaculture sites. So the other thing about this is that I want to point out is that, so these are all easy to use tools here, very intuitive. Once you give the Jayabots each their mission to go out, what you want them to do, you can also take them off that mission and operate them with remote control. So you could say, oh no, out here, let's take this one and let's go over here and start doing some dives over in this area. So you can do that while you're at the beach here. And so when you're very close by, within 500 meters or so, you can get real-time data from the vehicles through radio communications. And normally what we've seen most people use it is they'll send all 5 Jayabots back in, and then they'll do a high-speed Wi-Fi download of the data sets. Here's a picture of a Jayabot doing its profile dive. This part here is actually the bottom of the Jayabot, and it's gone down to the bottom and then taken the picture of what it's finding there. So there's a couple of really interesting development projects that we're involved in now. One is with the Aquatic Diagnostics Lab at Roger Williams University with Galit group. And we were just recently funded by the Rhode Island Commerce Corporation with an innovation voucher grant to fund a project together where we're working with them to automate the collection and filtration of water surrounding aquaculture sites. And the Aquatic Diagnostics Lab is extremely well known in this area. Everybody sort of sends their samples there for pathogen detection. And so we're trying to automate that process, keep people from having to go out into boats and very intermittently test their waters, but to make it a much more easy and accessible set of data for them to collect. And then the next thing that we've just actually learned this week was we were funded by the USGS, which is really going to help us accelerate the development of our bio product. So the USGS Next Generation Water Observing System funded us to build out the capability for water quality testing, dissolved oxygen, pH, turbidity, CO2 sensors on the Jayabot. So those are two great avenues that we're moving in these days. Here's a chart of Jaya's application areas and markets. And we have current users where you see these large green marks. So across academia, Department of Defense, US agencies like the US Geological Survey and NOAA. And then these smaller check marks that you see here are the areas where we have identified strong product market fit and have entry plans. And so we're now putting in place partners and funding to pursue these areas. This is a quick look at our current some of our current customers and the applications that they're using the Jayabots for. And I'm trying to keep within my time here. I'm going to forward this. So this is a look at our company. We're a startup company. We were founded about just a little under 3 years ago by Ian, Estefan Owen, and Jason Webster, co-founders. They've worked together before in other companies and in similar markets, and have put together a great engineering and production team and business development to start pursuing these other markets. This is, we don't count these as our full-time employees, but they're contractors. And in fact, each one of them has been with us for at least over 2 years, some from the very beginning conception of the company. So they're important members of our team. And our board of advisors, we work very closely with. Our board of directors and our advisory board, which we're starting to grow out. And we've participated in 3 different accelerator programs. So that's been super useful for us for company funding and also just exposure to different markets.
Thank you so much.
If we have any, yep, you're welcome.
If you have any quick questions, we are on time, but don't want to take any time from the next speaker. So if you have any questions, you either can contact Gyro Robotics through us or through their email. Okay, so I would like to introduce the Executive Director of BARD. BARD is the U.S.-Israel Agriculture Research and Development Fund, and I'm happy to present Professor Yoram Kapulnik.
Okay, I'm very happy to be here. I hope that my presentation is being shared. Can you see it?
We can see it. It's not in slide mode, though.
Okay. Okay, so again, good evening to you here in Israel and good morning to you in the United States. I'm very happy to be here. I'm the director of BARD, and BARD stands for Biological Agriculture Research and Developing Fund, which was initiated 45 years ago by the 2 governments. And our main mission is actually to support the collaboration between scientists between the 2 countries. Recently, we actually start to support or develop program that would fit to a collaboration between academia to industry. And this is something that I'm going to shed more light on it in a few minutes. Our main mission is, of course, to support beneficial, mission-oriented, strategic, and applied research and agriculture also in the food and nutrition sciences, and jointly that will be carried by the scientists from both countries. Now, in our portfolio, we have several tracks. Of course, the main one is the research project that we are supporting, but nevertheless, we have also feasibility studies. This is 1 year study for trying to show or to prove a concept. We have the postdoctoral fellowship. This is for a graduate student that actually receive a degree in United States and they are coming to Israel, or vice versa. We have a fellowship for graduate students to come to the other countries' laboratories, and also we are supporting workshop. This is a funding to get together dozens of people from each country and let them discuss some topic which is relevant to both countries' agriculture. Now, over the years, we supported a lot of topics that you are discussing here today, from fish and seafood health, algae and seaweed farming, aquaculture system, hydroponic and wastewater management, and of course, breeding programs for fish and seafood. But in my presentation today, I would like to highlight three tracks of application which have been developed in the last couple of years, and they are separate from the main program. They are addition to our program. The first one is the food and nutrition, which will actually the announcement will be in the first quarter of 2024, and this is for food and nutrition challenges. Again, collaboration between US scientists and Israel. Usually we are teaming very well with USDA's and NIFA, and NIFA scientists or NIFA's projects are welcome very much. Now, the next one, which again, the first quarter of 2024, this is really a new track. This is nutritional security. And again, we will appreciate any projects which are relevant to nutritional security. And the announcement, again, as I mentioned, will be very soon. The last system, which is actually was tailor-made for aquaculture and desert farming under the umbrella of climate smart agriculture. Again, every topic which is relevant to food from the sea and the desert, including fish, algae, and seafood systems. And of course, it can be inshore, offshore. All of those topics are relevant to this track of application. So I would be very happy to see application from both of you. I mean, from people from the US as PIs and also from the Israel, and the collaboration, of course, is appreciated. Thank you very much for your attention. And if you will need more information, please get on our website and you'll see that all this information is there. If you have any question, you can contact me personally. All my contact information is there on our website. Thank you very much.
Thank you, Professor Kapulnik. And I would like to thank all our speakers for this part of the conference, and I'm transferring it to Roni and Karen. Please take the lead for the next part.
Hello, everybody. Hi, Yoram. Thank you very much. We saved the best for last. Now we're really fast speed dating with the Israeli exciting industry. And our first, I'm thrilled to present Nissim Chen from Akwinovo, co-founder and CEO. Nissim.
Hi, everyone. As Roni said, I'm Nissim Chen, CEO of Akwinovo, and it is really a pleasure to be here today. And thank you for Roni and Karen for the opportunity to speak here. And Akwinovo is making more with less. We are, if you can go to the next slide, we have a solution that meets, can we see the next slide, please? Yeah, we have a solution that basically meets the growing demand for animal protein. And as we all know, there are three main tracks that are leading to that. The main the main and what we believe in the future to be the main source of accessible and cheap and natural animal protein is livestock and aquaculture. And we actually addresses the farmers' biggest need, as indicated in latest Alltech survey, which is digestion, which is gut performance and health, how you could improve and make a more efficient digestion. And actually, this is also the reason why, if you look on this market, it is rapidly stepping up, going to exceed $5 billion in 2027. So could you move to the next slide? Our technology is basically a platform technology that are based on biostimulants, which are fundamentally proprietary peptides, new molecules that have a unique mode of action that enhances significantly gut performance and health, and enable not only the novel products, but potential synergistic combinations with other existing products. It is designed based on naturally occurring peptides that play that fundamental role in gut physiology, and also stimulates and strengthens the immune system. And therefore, it improves biomass by improving FCR, creating more biomass with less feed. It increases fillet yield, survival rate, and could be used also to replace antimicrobial growth promoters. It was proven in large-scale studies in Asia with tilapia. No, go back, please, for a moment. It was proven back. One more back, thank you. It was proven in large-scale studies in Asia with tilapia and initial studies with salmon in Scotland, but it is applicable across species. And at the same time, we have generated, which includes all aqua species as well as terrestrial one. And at the same time, we developed the necessary component of production that will enable cost-benefit and scalable peptide production system. Creating more with less obviously has a sustainable effect on production system, impacting life underwater, hunger, poverty, and so on. Next slide, please. Yeah, and the market segments that we are approaching are all aqua species. As I said, tilapia would be our first product to be released in 2024. But we are also looking to develop products for terrestrial commercially important species like pigs and poultry, and even for pets, not treating biomass, of course, but treating gastrointestinal disorders that impact the life of pets. We are planning to manufacture our peptides with CMOs to sell it to animal nutrition and health companies who would incorporate it into the feed and sell it onwards to the fish farms. We are looking to have global strategic partnerships with many players, and we already have such a partnership with Archer Daniel Midlands. They are one of the largest players traded in the New York Stock Exchange. So as I said, we are planning to launch our first product for tilapia early 2024, add additional 3 products in 27, and additional 4 products in 29. Next slide, please. So to wrap it up, we have developed a platform technology that addresses the farmer's biggest needs, which is digestion and gut performance and health, and meet therefore the increasing demand for animal protein, using disruptive new bioactive peptides that are based on unique novel mode of action that secure strong and persistent effect, therefore the loyalty of the farmer, that includes target local activity in the gut. It was proven in tilapia and salmon, and we have the scalable and cost-benefit production system to allow mass production. It obviously addresses large and growing markets based on extensive IP and with a positive ESG impact that we have demonstrated. And we are now in round A, and of course, we are welcome many investors and strategic partners to join us in our journey. And that's it for today. Thank you very much.
Thank you very much, Nissim. Our next speaker is Eli Bergman, the CTO of BioClear Company. Eli?
Hi, hello everyone. Thanks for the introduction. My name is Eli Bergman. I am the CTO of NanoClear Water Solutions, which is the parent company for BioClear, our branch in aquaculture. And I'm very excited to present to you today our unique solution that is really revolutionizing the way that we treat water in the industry and solving some of the biggest challenges that recirculating systems are facing, as well as pond aquaculture. That is our ProBiomedia, which was a product of many years of development, more than 15 years of research by a team of PhDs who developed a nanomaterial technology which is extraordinarily porous and also has an extraordinarily high surface area per volume. And what does that do? That gives us a great growing area for productive bacteria that we use to treat the water. We are benefiting the microbiome by growing productive bacteria and removing contaminants. Now, what's exactly different about us? You can see a picture of our product in the right-hand side of your screen. And when we talk about biofiltration, the key is surface area per volume. Now, ProBiomedia, as you see, compared to a typical BioBall and the plastic bead media that we use in biofilters in the industry, has a surface area more than 500 times the plastic bead equivalent. And that amplifies the potential for productive bacteria growth and allows us to achieve, first of all, water treatment in much more efficient means. We're able to remove contaminants more quickly. But in addition to that, we're cultivating the microbiome. We're actually promoting bacteria in such a concentration that they are dominating the water, and they are preventing even pathogenic bacteria from coming in. Now, present some of our recent work, which was challenging to choose just one area that we've been testing in, but in fact, there was a very great result that we got at Auburn University in the States, and we found there, using ProBiomedia in a recirculating system in a tilapia grow-out, that two equivalent systems with a similar biofilter, the only difference being the use of ProBiomedia, and we get a 78% reduction in ammonia buildup compared to the control system, and 24% food waste, less food waste. The system ran more smoothly, and the fish were much more productive. And most importantly, we get a significant improvement in the yield of the system, as much as 20% improvements. We've seen that in tilapia, we see that in shrimp, we see that in other species. Just very quickly in work that we've done in Vietnam, we're seeing in shrimp hatcheries as well as in shrimp grow-outs that we're able to impact the bottom line of farmers significantly because of better water quality that we produce, and simply producing a healthier and more viable product. That's the really compelling aspect that we present. ProBiomedia is really revolutionizing the way that we treat water in the industry, solving some of our biggest challenges. It's allowing us to increase stocking densities. It's allowing us to simply outcompete disease in a very competitive space. We're able to remove ammonia very quickly, and we see that we are head and shoulders above a lot of competitors who are not able to manage ammonia nearly as well. And we find that to be one of the most standout qualities of the work that we do. And in RAS systems, where we're very concerned about effluents and we need denitrification, we're also able to provide that for our clients. And really appreciate your time. I look forward to potential partnerships, and please be in touch if you found something interesting and we can potentially work together. Thank you very much.
Thank you very much. Please, Yaron Bartal, Mariculture Systems, the CEO of Mariculture Systems. Yaron? Okay, Yaron,
Good evening, good day, everybody. Okay, Mariculture Systems, we are doing open sea aquaculture. Next slide, please. Okay, we are seeing a trend in the aquaculture world of shifting from nearshore fish farms to offshore large production capacity platform. That is either for ecological reasons, from tourism reasons, but people don't want the aquaculture nearshore. And the economies of scale makes it, I would say, necessary to have large capacity production offshore. It's essential either for us, the the owners or the regulator that has everything concentrated. We developed, not yet, please. We developed a system, a platform that has a fish production capacity of 8,000 tons per year. This is by having cages volume or gross volume to grow fish of about 300,000 cubic meters. And the uniqueness of that, or one of the uniqueness, is that it's the only platform that can withstand harsh sea conditions of over 17 meters waves. It's patent protected, and we are in the process of permitting the installation and production in several countries around the world. Israel is one of them. Next, please. So how does it work? You see the picture, the illustration on the top right. It's a semi-submersible, very sturdy rig. Nothing unique about that. It's floating in the ocean. It's anchored to the bottom. But the uniqueness is that around it, we have 4 large cages to grow fish. They are not connected to the rig itself. They are tied with the cable chains. And the uniqueness is that once there is a storm, we know that fish dive in deep water naturally. And this is exactly what we do here. We lower the cages to about 30 meters below sea level, and the fish continue their lives as if there's no storm. But you can see that in the drawing of the bottom right, in the upper position and in the lower position. We have automatic feed system. We have AI-enhanced growing factors or growing monitoring the fish with using an AI. And can you please go back one to the first slide? Thank you. Once the cages are leveled, except for harvesting, all the activities of the fish are remaining, all the monitoring, the control, the feeding, and so on. It's very sustainable. It's a self-sufficient photovoltaic or other renewable source energy. Those for most of the routine activities. And the 8,000 tons per year, that can be easily scaled up by having more and more platforms. And economically, it's high profitability, and thus the ROI is quite fast. There is a very nice video I don't have time here to show, but if you look for mariculture systems on YouTube, you can see that. Last slide, please. So what we're looking for, this is a seminar for the US market. We're looking for a strategic partner to support our penetration to the North American market, US, Canada, and so on. And like every startup, we're continuously seeking for investment. In the next slide, my contact details, I'll be happy to elaborate and to talk with anyone further. So thank you very much.
Our next speaker is Mr. Eran Adas, the VP BD of EcoShrimp. Hello, Eran.
Hi. So I'm the CTO of EcoShrimp. A little bit about the company. We have been operating for 7 years now. We are located in the south of Israel, and our mission is to develop and operate indoor and intensive systems for shrimp. A little bit about the next slide, please. A little bit about the industry. So the world production of shrimp is about 6 million tons per year, and actually it's the highest valued aquaculture product. The shrimp with value of more than $50 billion per year. So it's a very big and important market to act inside. Nowadays, most of the shrimp is produced in outdoor ponds, extensive ponds. Almost all the aquaculture shrimp belong to a single species, the Pacific white leg shrimp. And most of the production is carried out in around the equatorial belt, especially in Southeast Asia and in the South America. And the present culture method is considered by many to be unsustainable, and there are many reasons for that. I will mention only the key reasons. First of all, because it's extensive and technological level is low, there's very high levels of pollution, both organic pollution and microbial pollution produced by these farms that is released untreated to the environment. Because the traditional systems must be located close to coastal areas, there is a vast destruction of sensitive ecosystems like mangrove forest. Because it's impossible, because it's outdoor, it's impossible to keep those cultural systems biosecured, and therefore those farmers must use chemotherapy like antibiotics that are a significant cause of spread of antibiotic bacterial strains, harmful both to the environment and also affecting product quality, as some of the products contain residues of antibiotics that are consumed by men. Back 2 slides back. I'm another one back. Yeah. There's a very wasteful use of inputs of water and land, because the systems or the cultural systems are very extensive, and because the global production is strict only to the tropical areas, and the markets are mainly in the US and Western Europe, the cost of transportation and the pollution that are associated with this transportation is also very destructive. Next slide. So our technology is actually aiming to mitigate most of these problems that I just mentioned. We have developed an indoor system, intensive, that allows a culture of shrimp in very high densities, and in any place actually in the world. The systems are completely controlled, and they are not affected by any environmental conditions or climate. This allows us our system to produce all year round. We can build the systems close to the market, so this enhances the quality of the product, as well as limiting the pollution or the CO2 footprint associated with the transportation of the product. Especially after the corona, it was quite obvious to a lot of people that production should be local and rely less on long distance import of food items. Because our system is very intensive. Go back. Our system is
Eran, we are on a tight schedule. I'm sorry, but we have to move on.
Okay, so our system is a complete system. It's not it allows all the components that are needed to operate this system. That includes the design, the control system, the feed, the water treatment, everything that is associated with the production of the shrimp. And we're looking for partners that are interested in this type of business and want to join us and to invest with us, and trying to change the global shrimp production. Thank you.
Thank you very much, Eran.
We're a fully commercialized company. We do skincare products, starting from medical device down to cosmetics. And the material is also being used as a base gel for a therapist. It's a booster that enhances any kind of formulation. Next, please. The next slide, please. Why to choose Silaria? We are fully sustainable. We're the one before, please. We have a sustainable production that is repeatable year after year, and the material is proved to be with higher efficiency than any other extract. We have analytical proof that we are 10 times more in ingredients with active ingredients compared to an extract. We have clinical proved in several clinical trials our efficiency for medical unmet issues, for cosmetics also clinically tested. We did some of the projects with the Authority of Innovation in Israel. We received some backup, and today we work. I mean, we supply our products over the last 5 years commercially to companies in Israel and abroad. Next slide, please. What we're looking for, we're looking to expand. We're looking for distribution. We're looking for more clinical trials. And we do that in Israel. We have contacts that we're working with. It takes a lot of time. It takes the cycle for a company to join us is too long for what we would like to have. But it proves itself. We managed to show efficiency and to show that we are capable to comply with the needs of the skincare market. It is a huge market, more than $60 billion per year. That's it. Thank you.
Thank you very much, Noam. Our next speaker is Omer Polak, Program Manager at Zvia Coral. Hi, Omer, you can open your mic.
Okay, hear me? Okay, I'm going to present very simply. I'm a high school teacher, and I'm hearing what you guys are seeing. I'm representing something completely different. I just want to share with you what I'm doing, and maybe have some kind of a connection with you. The uniqueness of the product we have a small coral farm for high school students. And the purpose of doing that is having a holistic teaching for growing corals and teaching the students to have maintenance and also business development. It's like a multi-purpose grown teaching. We grow corals in captivity, and the purpose of growing at this stage is growing corals for the aquarium trade. Most of the corals that we get in the aquarium trade comes from the sea. And as being concerning the environment, we take care and we grow corals which are not from the wild. They also grow better. We grow corals without diseases and in high quality. And we're also starting a research and development to advance the breeding methods. Most of the breeding is by propagation, and we want to see and check it out. We have a new wall of high quality equipment. And the idea is to use this originality and the non-conformitism of youth to have new ideas and see how we can get new ideas. And hopefully these ideas are going to come into the industry. The initiative in general is called breeding. The students can be, part of the thing that we do is also most of you are growers, and from my experience, many of the people that grow, they don't know any part of the business part. And what we're doing is we're connecting the two together. So the idea of doing that is integrating the students with the growth process on one hand, and also learning how to sell what they grow in high quality. And the purpose of doing that is having kind of an external motivation to teach the students, first of all, to go and study. That's the main purpose. And the second thing is make them and train them into the future of perhaps being one of your employees. So we actually have many kind of goals. The main goals that we have for the educational purposes, we have entrepreneurship development, we develop life skills, we build self-confidence in the students that we have, provide tools for their adult life, study for the water market in general. We teach them everything basically that you need to know in at least the basis and to high quality. And we go and dive in the Red Sea. We teach them to conserve the environment. And hopefully these people that we're teaching right now are going to be in the future your employees. And what I actually want from you is, if you have any kind of way to get connected with us, if you want to come to Eilat where we are positioned, and if you want to give a talk, if we can come and visit you, we'll be happy to do that. And also, if you want to be part of the program that we have, contact us. It's quite easy to find us. Give us a call.
Thank you very much, Omer. Our next speaker is Michael Ada, the founder of Sapir Adir Company. Michael, you can open your mic.
Good evening. We are very happy to be here. I'm in charge of business development for Michael Ada, so I will speak now. So, well, I will present the Sapir Adir company. So the need is to find a cheap way to produce spirulina, the macro algae. So first, as a proteinic feed for fish in aquaculture to replace fish meal, something that is more sustainable, as biofertilizer, as the source of proteinic raw material for new vegan food. It is already used as superfood, but because of the expensive cost of this spirulina, it is just for currently human superfood. But if we succeed in to get to a lower cost, so even it can be extended to biofuel and store carbon. So why spirulina? First of all, the yield, 20 to 35 tons per year and hectare with 65% protein, as compared for soja here. So yeah, so it can be cultivated in alkaline water and that protect it from contamination. Also, it's fine also for saline water in a large range of temperature. We can have also the harvesting an easier harvesting because we need a filtering of 50 micrometer mesh, but it's not very expensive. And currently, as I've said, we speak about a market of $400 to $500 million, but mostly for human superfood because of the high cost of production. But again, with a lower cost, this can be multiplied. Next slide. So the problem. So what are the way to cultivate it now? So first of all, we have the sodium bicarbonate that is used as carbon raw material, and this represents 80% of the raw materials cost. That is $1,800 per ton of dry algae. There are other ways like use of CO2 flue that go out from chimneys and to try to use it, but it's not a viable option. Also to take from the air the CO2, but again, it is too expensive. Also, there is another experience from the US, the OE, for alkalophilic microalgae, but it's not suitable for spirulina because of the alkalophilic that is too high, the pH that is too high. So what is our innovation? First of all, there is a use of a proprietary technology that can be patent protected to capture CO2 from the atmospheric air combined with microalgae culture. So it can be implemented in regular paddle wheel driven raceway ponds with very limited investment, something like $50 per ton of dry algae. And for the raw material, we are below $300 per ton of dry algae compared to the sodium bicarbonate. So what is the status today? So the status is pre-seed. We have some interest from InNegev Technology Incubator, and they want to examine the idea. The technology's readiness level is TRL3. The technological complex were defined. The basic principle were observed. We already have a prototype in outdoor in real condition that can produce this spirulina. And so a 5-liter experimental and a 10-liter demonstration prototype. So, and you are invited to go and visit and see this with your eyes. Thank you for listening.
Thank you very much, Michael. Our next speaker is Omri Sharir from Bar Algae. He's Biz Development Manager. Hello, Omri.
Hi, everyone. Just one moment. Can you see me? Yeah. Okay. So I'm Omri Sharir. I'm the business development here at Bar Algae. I'll try to do it quick. And if you could go just one slide back, please. Okay, so Bar Algae is a precision microalgae company specializing in premium microalgae production. It was originally set up as a manufacturing facility for our mother company, which is TransAlgae, which specializes in genetic modification of microalgae. Since then, we've gone on our independent path, and we've set up the facility in 2018 and run since 2020, and we've been in the market for the last 2.5 years since COVID. You can move to the next slide, please. So Bar Algae developed a novel technology of cultivating microalgae in a very unique way, very pure strains, and in I'd say data-based manner. So you can see, I'll just mention some of the key differentiators of our facility. One of them is the ability to cultivate simultaneously different species of microalgae. Another one would be the very tight QA and QC. We have very strong reference to what is going on in the bioreactors in each segment or each part of the production and the cultivation. And another differentiator would be the fact that we are able to cultivate not only simultaneously different strains, but a wide variety of strains, from sweetwater strains to saline to hypersaline strains. And maybe one of our strongest abilities would be the breakdown and the actual cultivation of these different strains. We can go to the next slide. So at the moment, as I mentioned, we've entered the market, accessed it 2.5 years ago. Since then, we've been selling in 3 key markets: the nutraceutical market, the cosmetic market, and the aquaculture market. In the aquaculture market, our main subsector would be the hatcheries. whether it's shrimp, marine fish, or bivalves and oysters, which was mentioned earlier. So we are selling and we're producing and selling on a commercial scale for the last 2.5 years. As I mentioned, to different markets for different ingredients. We've been going up the value chain now for a while, whether it's specializing in unique formulations for the aquaculture market or offering high value extracts for the nutraceutical market, such as fucoxanthin, astaxanthin, different extracts of HUFAs and PUFAs and unsaturated fatty acids, beta-carotene, different extraction methods, whether it's supercritical CO2 or solvents, depending. A few, I'd say, side sectors which were already in the development are biostimulants and pet food. And in a moment, if you could move to the next slide, I'll mention another key project of ours, which is methane reduction. We've just been granted a couple of months back a grant from the Israeli Innovation Authority, and we have very good POCs presenting a premix which we've developed for ruminants, for methane reduction in ruminants, which is very promising, and we've been really developing it for the last couple of months with very good results. So as you can see, could you go back a slide, please? As you can see, we have, I'd say, 3 fields of interest, which we'd be glad to interact with our counterparts in the States, whether it's looking for strategic partners and distributors or customers in the different before mentioned fields of interest or the sectors we were mainly selling it to this point. In R&D, we're looking for interesting formulations and formulators who are willing to work with us. Please, could you go back a slide? And also doing some metabolomic work and analyzing microalgae. I didn't mention it, but another key differentiator of our company is we've already broken down more than 10 different strains of microalgae for commercial upscale cultivation. So we have quite a wide span and variety of different microalgae to date, which each of them, of course, has thousands of interesting compounds and secondary metabolites, which could be very interesting. And we're willing to work with the academia and different companies which find interest. And of course, we're also looking for funding and
Thank you, Omri. We are on a very tight schedule. We have 2 more speakers. So we'll move on to Kobi Levy, our next speaker from Fishways Company.
So good morning and good evening, everyone. Thank you for joining us. I know you guys are probably tired as I am the last speaker, but stay tuned. My name is Kobi Levy, I'm the CEO of Fishways, and today I want to talk to you about a groundbreaking innovation that has the potential to transform the landscape of sustainable RAS aquaculture. We at Fishways offer not just a solution, we offer a real revolution. In a world where the demand for fish and seafood is soaring, like you guys already know, Fishways offer a great solution to address the biggest challenge in the RAS aquaculture, the biofilter. At the heart of our innovation is the SBP capsules, a small bioreactor platform that is designed to replace the traditional biofilter completely. These capsules are a game changer. They provide a much more cost-effective solution. They can reduce capex by a staggering 50%. And on top of that, they can also save on energy, water, and skilled manpower. To illustrate the impact of Fishway technology, let me share with you a small success story that we have. We conducted our first proof of concept, and we grew 3 bream to market size without any biofilter besides our capsule, and with minimal water discharge. Thankfully, we had a remarkable success. thanks to the efficiency of the capsules. And this POC not only validated the effectiveness of the capsule, but it also showcased the economics and the environmental benefits of this innovative approach. Now, encouraged by this success, we're now on the brink of a new venture. We are now in these days constructing a semi-commercial fishery in Mitzpe Ramon in south of Israel that will focus on cultivating salmon, which we have already started growing, and sea bass using only the fish waste technology with no traditional biofilter. Now, as we look to the future, fish waste technology signifies more than just a mere solution. It's really a revolution in the RAS industry. And because of the many economical advantages that our technology brings, it not only paves the way for industry giants like today, but it also paves the way for a small and medium-sized player in the industry. In conclusion, fish waste is in the forefront of innovation, addressing immediate needs and setting the stage for a more sustainable and economically viable aquaculture industry. Thank you for your attention, and I look forward for any question or inquiries. We are looking for joint ventures and potential strategic partners. So thank you very much.
Thank you very much, Kobi. If there are any questions now for any of the speakers, you can either text them or ask them now. We are here to do any kind of connections. I can't end this event without mentioning 240 hostages in Gaza Strip. We are all praying to have them back safely. Thank you everyone for being here today. And this record will be online soon.
Yeah. Yeah. First of all, I would like to thank our team, Dr. Galit Sharon, Karen Almuzlino, and Ronnie Sussman for terrific collaboration. And I think it's a beginning. As we said in the beginning of the event, we're planning to have an online, in-person event June 3rd, and 4th here in Rhode Island, and we hope to meet a lot of you people that can come and invite other people from other countries as well, because I think what we saw today is there is a huge amount of opportunity to collaborate in research and business. I want to mention quickly the Bird Foundation. It's a United States government and Israeli government that give up to $1 million grant for commercialization. It can be an academic institution with a business, and the idea to promote business, and it's a, as I say, government programs. So with that said, again, I hope everybody will be safe, and thank you to all the supporters, the speakers, and we're looking forward to see you in person in June. Thank you again, and have a safe day.
Yeah, the last thing I would say before we close is that every one of you has the agenda. On the agenda, you have links to every single company and to every single speaker and to us. So in case you wanted to ask or make any communication between you and a company, we'll be happy to help with that in any way, both Avi, Ronnie, and Karen. And thank you very much. Have a great day and a peaceful night.
Thank you very much.
Thank you to the speakers.
Thank you very much.